Piotrowski visiting us. Tatjana, although has a Polish name, she's German, and she was raised near Tübingen, where she went to university. For her diploma thesis, which is like a master's thesis, she came here to San Diego, to Glenn Northcutt's UCSD, so she's familiar with Southern California. And there she studied receptors in the head of fish. And she didn't go so far away because for her thesis she participated in the big screen done by Yanni Nusla and Volhard. So got at the tail end of that, and so in the isolation of first few mutants, particularly mutants in the pharyngeal region. One of them is very famous, called TBX1, which is the gene mutated in DGeorge syndrome. So that's what she did as a graduate student, and later on as in her own lab also. But she's been at the University of Utah for 8 years, where she's an associate professor, and there she has pioneered the work on an organ of the fish called the lateral line, which we don't have, but fish do, and it's very useful for fish because it's a sensory organ. So here in the context of Embryology Club, you will all know that this is very famous because this was the first time that Ross Allison did a different transplantations by taking a very early tadpole of a pigmented salamander, cutting the head off like that, but then putting in and that was an albino tadpole, and then put the head of a pigmented tadpole, and found that there were cells that move all the way to the tail from the ear region. So that's how it was found that these cells migrate, and that, of course, the cells that were pigmented, so he could follow them. And with that technique, then Spemann did the famous organizer transplantation. It was the first lineage tracing was done on the lateral line. This is a classical system which behaves, as you will see, very much like an embryo. So thank you, Tatjana.
Thank you very much for the nice introduction, and also I feel lucky that I was invited here to speak at the Embryology Club. It's really an honor. So I also would like to thank the people who took the time this morning and this afternoon to meet with me and showed me the really interesting science. I really had a terrific day so far. So let's see, this is the pointer, I think. Yeah, so here what you're looking at, so my lab is interested in as Eddie explained, in the development of the lateral line system, and also in how the hair cells that are the sensory organs in this lateral line system, how they regenerate, and I'll tell you about this during the talk. So what you're looking at here is a fixed embryo, so this is maybe a millimeter long, and each of these green circles here is one of these sensory organs that has hair cells in it. All aquatic vertebrates have the sensory system, and they use it to detect water motion. And I think you can see this here, and this is a schooling swarm of sardines here. So this enables them to measure or to feel whether there are other fish next to them. Fish can also sense how fast water is flowing if they're swimming in a river. And they can also sense whether prey has fallen into the water or whether there are obstacles in the water. So it's really a sensory system. It's like touch at a distance that we don't have as land vertebrates. So the sensory lateral lines, or the lateral line system, was discovered or first described here already in 1664 and 1678 here by Lorenzini and by Sten. And what they described were actually these electroreceptors here in a shark. You can see here they're called ampullae of Lorenzini. And they're actually also part of the lateral line system. So the lateral line system consists of electroreceptors as well as of these mechanoreceptors that detect water motion. So here, this is a primitive ray fin fish, and here I depicted where all these receptors are located. So these little dots here are the electroreceptors, which are located in the skin of the fish. Then these lateral line sensory organs can be located here in the skin of the fish. So the sensory cells project directly into the environment, or the lateral line sensory organs can be housed in these bony canals. And if you go to an aquarium or to a fish market and you look at big fish heads, you can sometimes see holes around their eyes, and these are actually the openings into the bony canal system that houses these sensory cells. And so the functional unit of these sensory organs here are these hair cells. So each of these green circles that you saw in the first image here is one of these sensory organs. And it consists of a number of hair cells here in the middle. And they're surrounded by different types of support cells. So there are these inner support cells and the outer mantle cells here. And here in this electron micrograph, scanning electron micrograph, you can see the cilia sticking out into the water. And now as water flows across these cilia, they get bent and they open up ion channel mechanically, which then leads to an action potential here in these hair cells that travels then to the brain. And then what's important to note also is that these hair cells are very similar to the hair cells in our inner ear. So mutations that we found here in zebrafish that affect the lateral line system have been shown to also cause deafness in humans in the hair cells in the inner ear. So I think even before Harrison here, Julia Platt at least described how this lateral line system developed, even though she did not do any functional studies. So Harrison was the first one who did transplants. So this is a mud puppy here, embryo, and you can see here this placodes migrating down from the head region here towards the tail tip. And during that migration, this placode deposits clusters of cells which will form then these sensory organs. And so this is here a depiction of how this looks like in the zebrafish embryo. So within 20 hours here, the placode forms behind the ear. So the aortic vesicle would be here. It migrates towards the tail tip. And here the red dots represent the sensory organs which will form.
Tatjana, is this part of the ear placode or is it a different part?
It's a different placode. So the aortic placode is different from the lateral line placode. And so there was originally the theory that the lateral line system was more primitive and it gave rise to the ear. However, what people nowadays think is that these are two independent sensory systems, however, which are derived, where the sensory neurons basically are derived from ciliated neurons, which then develop into an ear and the lateral line.
And which cranial nerve innervates them?
So these are they're innervated by lateral line nerves. And they're independent of the classical cranial nerves. So they enter the brain via separate rootlets and also project into different nuclei in the brain. So they're really separate. However, they can fuse with some of the classical nerves, and that's why people sometimes thought they were actually part of some of the classical nerves.
Does the whole structure require have a common precursor or progenitor like sensory organs in the fly, or does it have has neurocrest input as well?
So there was one study by Andres Collazo actually here in UCLA who thought that neurocrest cells can also contribute to the lateral line system. But then many other studies say that it's purely placodally derived, and that this placode gives then rise to hair cells as well as support cells, as well as the ganglion that stays behind and the axons that innervate the system. So I think this probably needs to be revisited to see whether there's any neurocrest contribution, but if there is, it's probably small. So what you're looking at here is the trunk of the zebrafish, and you can see the muscle somites under here, and this is the epidermis, and this is the migrating placode as it migrates towards the tail tip. So you can see here it leaves behind single cells. In this movie, I didn't catch a process where it's depositing an entire sensory organ, but look at this cell here, it'll divide. And so this really depicts here, or shows that we can watch now here the placode migrating in a live fish by this particular embryo was stained with a fluorescent dye, a lipophilic dye that labels all membranes. But you can see this really with the IC optics in the live embryo. So this is a transgenic line with Claudin GFP, which is a tight junction marker which labels the lateral line really nicely. And you can see here the primordium is ready to deposit a sensory organ, a neuromast. And then here the next one forms as this one gets deposited. And again, there's a number of cells here that are being left behind here as well.
So how deep into the organism is this?
So this is right beneath the skin on top of the muscle. Yeah. And this is just a lower magnification movie of the same process, and I think it shows you one or two of these, what we call deposition cycles, where one sensory organ here gets ready to be deposited. And you can imagine, as the primordium migrates, that it actually, since it's losing this many cells, that it actually has to make up for the loss of these cells. And there is a lot of proliferation going on during the migration of this primordium.
What does it migrate along?
So here in zebrafish, it migrates along the horizontal myoseptum, which is a connective tissue, and there are also slow muscle fibers associated with the myoseptum, and they secrete a chemokine called SDF1, which I'll show you later in the talk, which basically guides the primordium. So this is just a summary of the movies I've just shown you. So the primordium migrates towards the tail tip, and then after 20 hours or so, you get here what we call the primary sensory line here. And here this is a magnification of the primordium or placode here, and you can see the sensory organs that are forming here are sort of forming in a rosette-shaped form, whereas here the tip seems to be unpatterned. And so we think that actually there are probably stem cells here located in the tip of the primordium. So this is, you can see here this depiction of the deposition cycle. So there are 3 rosettes here, then one gets deposited, and here these cells basically now, there's actually proliferation everywhere, but there is a new rosette being formed here in the tip as it migrates. So what we did in this experiment here is we just randomly labeled a few cells here by transplantation. So you can, in zebrafish, you can inject the donor embryo with a fluorescent dye. Subsequently, all cells will contain that fluorescent dye, and you can pull out some of these cells, stick them into an unlabeled host, and then you can watch these cells. And so what this is supposed to show you here is that cells that we labeled or that ended up in the tip of the primordium during migration are located more and more trailward. So there's this sort of progression from the tip towards the tail. Then in the movie, I also pointed out that the primordium deposits here these lines of cells here. And what we discovered in earlier studies is that these cells are pluripotent lateral line stem cells that post-embryonically proliferate and make more of these sensory organs. So in the primary lateral line, you have 6 or 7 of these sensory organs, but after 2 or 3 weeks, you actually have 20 or 30. So in every somite boundary, you'll have one of these sensory organs. And yeah, you can see this here with an in situ that labels the lateral line. So these are the sensory organs that have been deposited. In here is the string of cells, and you can already tell that here these cells start to proliferate and make more of these sensory organs. Then there are more, so we call these at least pluri- or multipotent lateral line stem cells, but we know there are also stem cells here in the deposited neuromass because throughout the life of the embryo, hair cells are being replaced in that sensory organ, arguing that there have to be stem cells in here.
So you may be coming back to this later, but if not, how does the placode know where to leave cells?
Yeah, so I will come to that later, and yeah, maybe I'll leave it at that because it's going to be the last part of my talk. So maybe I should, so you don't have to wait too long. So so there is no particular position that tells the neuromass where it should be on which somite number, but it's proliferation in the primordium that drives cells out. So if the primordium reaches a particular length, it starts dropping off. And depending on how quickly that happens, that's where the sensory organs are located. But they don't care whether they sit on somite 5 or 6 or 7 when they're. So this is just a summary of the introduction. The primordium, the cells in the primordium migrate collectively. There are, we think, 3 types of stem cells: adult neuromass stem cells here in the deposited sensory organs. There are these interneuromass stem cells and possibly tip stem cells. And we're currently trying to figure out where these stem cells are located. And then once we identify them, we would like to study the signaling interactions that maintain them and promote self-renewal. So these are the major questions we're asking in the lab right now. So how is directed migration controlled? How is this migration, patterning, and differentiation coordinated to form a proper morphogenesis in the lateral line? Where are the stem cells? How are they maintained? And how is hair cell regeneration controlled? And so first I'll tell you a little bit about neuromass development, stem cells, and regeneration. And this is still rather a project in progress, but I still would like to show you what we're up to here. So this is again the same image that I've shown you before, and just to remind you here, so the hair cells here are in the middle and they're surrounded by these support cells. Now, and in contrast to all lower vertebrates, mammalian hair cells don't regenerate. So as we age or kill our hair cells with noise, for example, loud noises, these hair cells will never come back and regrow death. However, in all lower vertebrates, such as chicken, birds, reptiles, amphibians, these hair cells continuously regenerate. And so we're aiming to identify genes that are involved in regeneration here in the zebrafish, and then at some point maybe we can go back to the mammal or and investigate why this process is not happening in mammals. So this is a mouse cochlea. These are the different rows of hair cells. So this is a healthy one, and here you can see where the hair cells were dying. So there's one inner row of hair cells which we're using for hearing, and then here these 3 outer hair cell rows. And you can see here basically that this animal lost a number of these hair cells. So here in zebrafish, we can kill the hair cells by bathing the embryos in an antibiotic. We are using neomycin, and this is actually also happening in humans. So if we take high doses of antibiotics, we can also lose our hearing. So, but what's happening in these zebrafish embryos is that 16 hours afterwards, these hair cells are coming back, and they're labeled here with a fluorescent dye that we just add to the water in which they're swimming, and the hair cells are taking it up. So this is a fairly easy screen for us to do by just soaking the embryos. And then we're looking for embryos which do not regenerate, and I'll show you an example later. So in order to understand regeneration, of course, we need to know which cells are giving rise to hair cells and where are the stem cells in the system. So this is a cross-section through the sensory organ here. In the middle are the hair cells that are dying. They're very likely sending signals here to these inner support cells as well as to the outer support cells. In response to hair cell death, these cells here start to proliferate and start replacing the hair cells. But the support cells do not only proliferate during regeneration, but also during normal homeostasis. So here we just took an intact embryo that was not manipulated, soaked it for 40 hours in BRDU to detect any mitotic cells, and you can see that a lot of these cells here now took up the label during these 40 hours. So now we really would like to figure out which ones are the stem cells. So there are these 2 putative stem cell compartments here, these inner support cells as well as the mantle cells. And right now we believe that probably these mantle cells are good candidates for the stem cells. And there are several reasons for this. So already old studies here in 1993 by Jeff Corwin's lab have shown that if you amputate an axolotl here and you amputate the tail tip, you'll also lose some of the sensory organs. Now, as the tail tip regenerates, the sensory organ closest to the site of wounding sends out mantle cells that start to proliferate and basically regenerate these sensory organs on the tail tip. So certainly these mantle cells seem to are multipotent in the sense that they can give rise to new sensory organs. Also, stem cells very often do not proliferate as much as other cells. And what you're looking at here is in green are actually labeled the mantle cells, and you can tell that they did not take up BRDU here, even during normal, during the normal life of the embryo here that was not manipulated. We also performed some label retaining assays. So this is a primordium here, which has the photoconvertible dye CADA, or expresses this photoconvertible dye CADA specifically in the lateral line. When we photoconvert single cells here, we found here, for example, if we label this particular cell here and then watch it over time, that it actually did not dilute the label much, and it ended up being a mantle cell. So, however, if we now treat these embryos or these neuromasts with neomycin, so this is a control embryo, and I apologize, there is no nuclear blue label here in this particular image. But the point I want to make here is that there's only one proliferating cell here. So this was a shorter BRDU treatment just for an hour. However, post-neomycin treatment, many of these cells start to proliferate, and even some of these mantle cells start to proliferate. So now, the approaches we are taking to identify these genes that are involved in hair cell regeneration are the following. So the first one is we're doing a gene expression profile of these mantle cells here in blue. And we're lucky in the sense that Vladimir Porsche in Singapore identified an enhancer trap line that expresses GFP specifically in these mantle cells here. And so what we're currently doing is dissociating these, well, first treating them with neomycin, killing the hair cells, then fact sorting these embryos, and then do microarray analyses on mantle cells from control as well as from neomycin-treated embryos. And I wish I could have more results on this already, but we're still just doing the arrays at this point. Now, the second approach we're taking is to screen for mutations that affect lateral line development and or that affect regeneration here. And again, this is one of these fluorescently labeled embryos here, where we then kill with neomycin. And we found one mutant here that looks very promising in the sense that the hair cells do not regenerate in this particular embryo. So we also quantified this here. So this is the number of hair cells here in a control embryo, and here as well is in wild-type embryos. In the black bars in here in purple are the mutants, and you can see they are making a few hair cells, but they do not recover as well as the wild-type embryos. And we're currently mapping this mutation and hopefully have our first gene that's involved in regeneration. So now in the second part of the talk, I'll tell you a bit what we've learned about morphogenesis of the lateral line, and specifically how the Wnt beta-catenin pathway is involved in that. So in addition to screening for mutants, we also did a candidate gene approach by basically we obtained
Yeah? Sorry, I missed something. How do you distinguish between a mutation that affects, let's say, the lateral line only or regeneration only without it being a general mutation?
Yeah. So that's a very good point, and it requires a lot of secondary tests to see whether. So the first thing we need to do is do these BRDU incorporation studies to see whether BRDU is downregulated in the entire embryo, right? So if the fish is sick or affects cell proliferation for some other reason, then of course there will be no regeneration. So yeah, so certainly I oversimplified by just showing this one image. So we have to do many more tests to really show it's regeneration specific for the lateral line. Other tests people do is...
that mutation you showed is regeneration specific?
We don't know yet. We think so because the embryo looks very healthy. So it's not one of these embryos that slowly die or that just look very bad. Other tests you can do is you can cut tail tips, see whether the tail tip regenerates normally, which could be done too. So here, now coming back to the second part of my talk here, so in addition to screening for mutations, we also did a candidate gene approach by requesting mutations that have been identified to be in particular signaling pathways. And Hans Klever's group in the Netherlands sent us a mutation here in the Wnt signaling pathway, and when we looked at the lateral line phenotype, we were pleasantly surprised here to see that the primordium didn't migrate properly here, so it stalls here in the middle of the trunk. It also leaves behind the smear of cells instead of nicely spaced sensory organs. And so the first question we asked is, so why does the primordium not migrate in these APC mutant embryos? And I should point out that APC here, a mutation in APC constitutively activates the Wnt pathway, so there's too much Wnt signaling going on in this embryo. And this mutation in APC is also the very same mutations that about 80% of all colon cancer patients have in humans. So when we look at the primordia in these APC mutant fish, we see that the cells are motile. So the primordium does not stop migrating because the cells lose motility, but rather because there's a lack of coordinated migration into one direction. We call this a tumbling behavior. So this is just an introduction, or maybe a reminder here what the Wnt signaling pathway is doing. So this is a cell here that in the absence of a Wnt ligand, so there's no signaling going on, or here in the presence of an inhibitor. And what happens here is that APC here is part of a protein complex here that targets beta-catenin here for degradation. So if this complex is allowed to assemble here, beta-catenin gets degraded and it does not go into the nucleus, and there's no Wnt targeted gene activation. However, in the presence of a Wnt ligand here, this complex of APC and these other proteins here gets disassembled, beta-catenin accumulates in the nucleus and then, or in the cytoplasm rather, goes into the nucleus and can turn on these Wnt target genes. And this is the situation that we have in the APC mutant, where the APC gene here is mutated. So there's too many Wnt target genes are activated. So Andy, the graduate student who is working on this project here, then looked where the Wnt target genes are actually expressed, and he found that they're specifically here localized to the leading edge of the primordium here, which is sort of this unpatterned region here. And not surprisingly, in the APC mutants, you can see that now these Wnt target genes are expressed in all cells. And yeah, in all cells. However, here, when we looked by actually performing completely independent experiments where we manipulated another signaling pathway, this time the FGF pathway, we found that we can phenocopy this migration phenotype very nicely. So up down here, as a reminder, this is the APC mutant. And this is here an embryo in which we blocked the FGF pathway with this drug that inhibits the FGFR1 receptor. The primordium also tumbles. You can see that here in this movie. So the cells are also not migrating forward in a collective manner, indicating that these two signaling pathways might be acting together to ensure proper directed migration of the primordium. So when Andy looked where the FGF ligands are expressed, he found that they're also restricted here to this leading edge of the primordium. However, what was a little unexpected is that the FGF ligand expression depends on the Wnt signaling. So in the APC mutant, where we have too much Wnt signaling, you also get too much activation of the FGF pathway. And in the heat shock dicop fish, so these are transgenic fish that express this inhibitor of Wnt signaling. If we turn that on and shut off the Wnt signaling pathway, we also lose FGF signaling at the same time. So the FGF ligands are actually diffusible molecules. So here's the ligand, and you could imagine that it could diffuse to these other cells. So in order to see which cells can actually respond to this FGF ligand, we looked at the expression of a transcription factor called PA3 here, which is a readout for in which cells FGF signaling is active. And we saw indeed that cells, even in the back of the primordium, respond to this FGF ligand, which is only made here in the tip, but which then can diffuse into the back. Surprisingly, we saw that the cells that make the ligand actually do not turn on the FGF pathway. And so this sort of indicated that there must be an inhibitor in this area which keeps these cells from responding to the ligand that they made themselves. And so we performed basically a candidate gene approach where we looked at many different FGF inhibitors that are described in the literature. And we found that Ceph, this transmembrane protein here, is also expressed in the tip of the primordium. It's able to turn off FGF signaling, and it is surprisingly a Wnt target here. So if we take away Wnt signaling, we also lose this inhibitor of FGF signaling. And you can see this here. So this is the wild type expression again of this P3 gene here as a readout for FGF signaling. If we now knock down or take away Ceph function by injecting morpholinos, you now get expansion of FGF signaling into the leading edge of the primordium. So Ceph is the inhibitor that keeps FGF signaling to the posterior of the primordium. So you end up here with this model here, or I guess we start with this model here now. So you have Wnt signaling is active here. It turns on FGF ligands, which diffuse towards the back. At the same time, it makes this inhibitor to ensure that this region here does not respond to FGF signaling. So at this point, we asked whether FGF signaling itself might also help to restrict Wnt signaling to this anterior end, and it does. So these here are again in wild-type embryos the two Wnt target genes. And these are embryos in which we inhibited FGF signaling, which is in the back, and now we get expansion of Wnt signaling into the entire primordium. So the question then arose, so what is the inhibitor here that keeps Wnt signaling towards the tip? And again, by candidate gene approach, we found that DICOP1, this diffusible inhibitor of Wnt signaling, is expressed in this area, and it is actually a target of FGF signaling. So FGF signaling here in the back turns on this gene and keeps Wnt signaling over here. And this just shows again that DICOP is an FGF target because we lose it if we get rid of FGF signaling. So this now provides us with this model here. So you have the migrating primordium basically is divided into these two major signaling pathways. So the anterior expresses the Wnt target genes, and the posterior here expresses the FGF targets. At the same time, these two signaling pathways make the inhibitors for the other pathway to keep these two pathways in separate areas. So this is a really nice model, and now the question is, so what kind of biological significance does this polarity have? And it has something, so each of these pathways has its specific function. So first, let's look here at FGF pathway. And it has also been shown here by these other groups that FGF signaling is actually important for making the sensory organs or for making these rosette shapes. So in a wild-type embryo, you can see here these little puncta in the middle, and this is actually the center of where the rosettes form. If we inhibit FGF signaling, you lose these puncta, and there are no rosettes forming. If we do an in situ for hair cell markers, which actually should come on in little dots here in the middle, we also lose them. However, if we look at an APC mutant embryo that has too much FGF signaling, here even the tip is full of FGF signaling, we now see sensory organ formation in the entire primordium, even in the tip of the primordium. And so this is the reason why you want to keep out FGF signaling, or you want to restrict it to the back of the primordium, because otherwise you actually you deplete your stem cell compartment if you make a rosette here and differentiate that into a sensory organ. And then you stop migrating as well. So we then asked, so does Wnt signaling here have an additional role other than just inducing FGF signaling in the back? And it has something to do with the migration of the primordium. So why do the APC primordia tumble that I showed you at the beginning in the movie? And so it's now pretty well understood here that this chemokine signaling pathway is the principal guidance system that is used by the primordium here. And let me introduce here the different factors. So this is SDF1 expression, which is this chemokine ligand that's diffusible, and it's expressed exactly here along the myoceptum, on which the primordium migrates. So it has been shown that if you get rid of this expression of SDF1 and you misexpress it somewhere else, the primordium will home towards these new SDF1 expressing cells. So it's a true chemokine signal here. In many systems, for example, if you look at cells in the brain that migrate towards particular nuclei, they will home towards the highest source of SDF1. So very often SDF1 is expressed as a gradient in an organism. However, here along the lateral line, it doesn't seem to be a gradient, because we have mutants in which the primordium can turn around on itself and migrate towards the head region, arguing it's not a protein gradient here of SDF1. Now there are two receptors that respond to this SDF1 ligand here. This is CXCR4B, which is more localized here in the tip of the primordium, and CXCR7B, which is here in the back of the primordium. Now it was shown that irrespective of which receptor you deplete, you'll disrupt primordium migration. And this was initially a little puzzling, because if you think about that, you have two receptors here that bind the same ligand. Why should they be expressed in different cells, or why is it important for them to be in different cells? So a clue came from Aris Raz's group, who works on germ cell migration in zebrafish, and what he's shown is that CXCR7B binds to SDF1, but it doesn't lead to an intracellular signal. It basically acts as a sink for SDF1, but doesn't do anything with that ligand. So what this could be doing is that it's actually making a virtual gradient of SDF1 within the cells. So this is depicted here. So this is the CXCR4B expression in the front. This is 7B in the back. Now here, this is the new nomenclature now. So this is SDF1 mRNA, which is ubiquitous along the myoceptum. Now, however, we think now that the CXCR7B protein here in the back actually creates a sink for SDF1, and there makes a gradient of SDF1 protein here in the primordium. And thereby, each cell in the primordium still sees a gradient of SDF1 and knows which direction it should go. So here, this is a movie in which the SDF1 stripe is disrupted. So the midline would actually be up here. This is the pronephros that you're seeing here. And you can see here that the primordium turns around and doesn't really know where to go. So this is what happens if you get rid of the SDF1 stripe. And this is a mutant here which affects somite boundaries, and the SDF1 stripe is not continuous. So I wouldn't have shown you here these chemokine receptors if they weren't affected in these APC mutants. So this is a reminder of the wild-type expression here, and this is the APC mutant down here. So CXCR4b is now expressed in every cell, and CXCR7b here is completely lost in the APC mutant. And this argues that Wnt signaling is repressing CXCR7b here in the front. And if you ectopically express it in the back, you get rid of the cells cannot perceive an SDF1 gradient anymore and start tumbling. So of course, we wanted to know where the Wnt ligand was coming from, and Rob Duncan in the lab, a graduate student, he cloned, I don't know, 24 different Wnt ligands to find the one that's expressed here in the tip of the primordium, and we found some very strange at that point expression patterns. So he found that Wnt9b is surrounding the primordium, so it seems to be responding to the primordium as it migrates by, and we're currently trying to figure out what that gene might be doing. Then we found a Wnt2 that's expressed in the posterior of the primordium, at which point we got a little worried about our model. However, luckily, when he did one of the very last in situs here, he found that Wnt10a here is expressed in the tip of the primordium. And it looks very much like, by doing functional analyses right now, it looks very much like this is the Wnt here that's coming from the tip region. Now, of course, the fact that we have Wnt2 here in the back poses new interesting questions, which we're trying to figure out right now, how that fits into our model, having a Wnt also in the back.
Do you have some kind of like top-gal model for looking at Wnt activity?
Yeah, so there's a top GFP line. Unfortunately, even though it's a destabilized GFP, it doesn't turn off quickly enough, so it's actually in all primordium cells.
Wasn't the Wnt2 just in a different, wasn't it just around the periphery of the...
That was 9b, okay. So there was even a third one.
Oh.
Yeah, and so I mean, it's such an interesting expression pattern, but we have no idea at this point what it might be doing, why the primordium turns on Wnt just to the side of it, and then what that will do. Yeah? Mm-hmm.
From your slides with the APC mutant, it looked that the number of cells cells looked smaller and also more numerous. Now, thinking that Wnt in flies, and I assume Zebrafish the same thing, also has a profound effect on proliferation. You mentioned earlier, right? The same zone, which here is purple, and it's like the Wnt. The expressing zone, but it's also the region which you previously thought has stem cells. So there's proliferation going on. Yeah. So my question is, so how do you kind of disengage these two processes of migration and directed migration and proliferation?
Yeah.
But because, so first of all, is it true that in this APC mutant there are many more cells, or is that just...
It is true, yeah. So we didn't know this at this point. So this was basically the first part of the study, and then we tried to answer exactly this question, and it actually all ties in together. So Wnt does actually control all these processes and basically drives morphogenesis or coordinates morphogenesis by coordinating migration with neurogenesis with proliferation that drives then deposition. And I'll show you this here in this talk. Yeah. So this is now exactly this question: what other roles might Wnt beta-catenin play? And of course we were inspired by your work in flies. And we're trying to figure out now where the stem cells are and whether it correlates in any way to the hindgut or midgut in flies, right? Where the stem cells are and what regulates them. So the third part of the talk is now how Wnt beta-catenin influences neuromass deposition by controlling proliferation. And so the first thing we wanted to know is what drives these periodic deposition? What tells the primordium, that the question we had at the beginning, what tells the primordium where to put down these sensory organs? And the first question is this determined by maybe the number of somite boundaries that it traverses, and this was postulated in the literature before, or is it primordium autonomous? And we believe now strongly that it's primordium autonomous. So this is one example why we believe this is, if you so here we're looking at a trilobite mutant, which has a mutation in a planar cell polarity gene, which affects convergent extension during early development. So the somites are skinnier here. So the primordium nevertheless, the primordium traverses the same distance before it drops off the next primordium, even though it has crossed now many more of these somite boundaries. So we quantified this, so the distance is basically the same. However, there are many more somite boundaries here. So it's not determined by the number of somite boundaries that the primordium traverses. So this is basically the questions we're asking. And then these were some hypotheses we had as we went into the study. So what could tell the primordium to periodically deposit these sensory organs? And in analogy to somatogenesis invertebrates, it could be that they're oscillating clock genes in the primordium, and as they sweep through the primordium, some of these cells fall out. Or it could be stable gene expression domains, and simply by adding more cells, you drive cells into a particular gene expression domain that tells the cells to now be deposited in. And I hope I can convince you that this is the case here in the primordium. So the fact that that cell number or cell proliferation in the primordium might play a role in deposition here came from the observation that the length of the primordium fluctuates during migration, during the deposition cycle. So here, this primordium is about to be ready to deposit a sensory organ, and it's basically this length. Then it deposits and it shrinks to this length until it proliferates again, until it becomes this length, and then it starts to drop off the next primordium. So we did several experiments to test that hypothesis that cell number affects deposition. And the first one is here. We looked at mutants that affect cell death in the nervous system. So this is a mutation in BAP28, which is a ribosome synthesis gene, and that has been described to cause cell death. And you can see that there is tunnel staining, which labels dying cells in the primordium. And now if you look at the neuromass number, you can see there are fewer neuromass, and they're also more widely spaced. We've recently also started doing experiments with drugs that affect cell proliferation, and we can get the same effect here. So by simply reducing the proliferation rate in the primordium, you get fewer sensory organs, even though they still have the same size as in a wild-type embryo. Yeah, so this is just showing that this is really cell death specific. So this is the wild-type number of neuromass on this side here in blue. And the mutant only makes 2.5 of these sensory organs. Now, if we inhibit cell death in this mutant by injecting a morpholino against P53, which is supposedly inhibiting cell death, or does inhibit cell death, we can rescue the phenotype again. So now the question is, so what are the signaling pathways that drive proliferation in the primordium? And when we looked at proliferation patterns, the first thing you see is that, so these are again BRDU cells, you can see that they are sort of restricted or mostly happening here in the anterior 2/3 of the primordium. And this is overlapping here with the active, whether the Wnt pathway is active. So we looked at different Wnt target genes, and at least some of them have a little more longer expression domain, which overlaps here with the proliferation pattern of dividing cells. And we wanted to see whether, since we were in search of a stem cell compartment, and we had postulated that maybe the stem cells would be here in the tip, we looked at proliferation patterns in these different domains. So this is the unpatterned region, and this is the first rosette, and the second rosette, and the third rosette in the primordium. And a little bit to our surprise, we found that the proliferation index here, or mitotic rate, in the anterior 2/3 is not different from between any of these domains. However, these domains here differ with the last one. So the last rosette is the one where the proliferation rate really drops down. So is the Wnt pathway controlling proliferation? And so we started looking at all the different manipulations that we have in hand. So this is the APC mutant. And as Volker has suggested, there is actually more proliferation here in these APC mutant cells. However, I should mention that we actually do not see an increase in proliferation in the domains where cells are proliferating in a wild type, but it's just actually an extension of an ectopic expression of proliferation in domains that usually do not proliferate. So this argues that even in a wild type, these cells here are probably proliferating at the highest rate that they can. So also, if we knock down Wnt signaling using this heat shock tool, again, now we lose proliferation significantly. Now, however, these two tools here also manipulate FGF signaling at the same time. So they didn't really tell us whether it was Wnt signaling or FGF signaling or both. And what we found here when we now knocked down FGF signaling with this pharmacological drug, what's happening in this particular embryo is that we knock down a Wnt signaling, but FGF signaling is high in these primordia. And still FGF signaling, there is no FGF signaling in these primordia, but Wnt signaling is high, and still we don't get proliferation here. So Wnt signaling, even though it's required, it's not sufficient to drive proliferation in the absence of FGF signaling. And vice versa, unfortunately, I don't have the data here. When we drive FGF signaling with a constitutively active FGF receptor transgenic, you drive FGF expression, you lose Wnt signaling, and again, we don't see an increase in proliferation, and we also see a loss. So we really need FGF signaling and Wnt signaling for proliferation to occur. So neuromass deposition is periodic. Does the domain of Wnt beta-catenin activation or the pathway activation oscillate? And so we wanted to measure the domain of the Wnt pathway activation here. And we used a tool which I've shown you before, is the CXCR7B expression, because we know that it's repressed by Wnt signaling. So basically all the empty domain here is where Wnt signaling is active. So what we now did is we counted, or we measured here the length of the Wnt pathway activation domain during different phases of the deposition cycle. So as the primordium grows and gets smaller. And we found here that the CXCR7B freeze domain, which is the Wnt pathway activation domain, is constant irrespective of the primordium size. So here in green, this is the length of the primordium here. And in red, this is the free zone. And you can see here by the slope of this graph that there's no correlation between how long the primordium is and how long the Wnt pathway activation domain is. So this argues that probably the Wnt producing cells here in the tip create a stable morphogen gradient that doesn't fluctuate with primordium length or deposition. And this is the control here, where we look now at the CXCR7 domain, and the CXCR7 domain nicely correlates with the length of the primordium. So the longer the primordium gets, the more CXCR7 positive cells you get, and this is the domain that does not see Wnt signaling. So now we would like to do the same thing with the FGF pathway, since we know that both FGF and Wnt are responsible for proliferation. But our feeling is really that these genes are not oscillating in the primordium, as you would see during somite segmentation, for example. And so this is the model we have for deposition. So the green cells here are the cells where you have Wnt signaling and as well as FGF signaling. And even though in the first half of the talk I said that there was absolutely no FGF signaling here in the tip of the primordium, there's actually a low level of FGF signaling present, which explains why there are proliferating cells in here too. So as these cells here proliferate, they displace cells here into this Wnt free domain. They turn on the CXCR7B gene, and then these cells get deposited. And so now, as a summary really here, I would like to say that Wnt beta-catenin signaling, there is coordinating morphogenesis by really coupling the migration via the expression of these chemokine receptors. It couples it with neurogenesis by keeping FGF signaling in the back, and it also couples it with periodic neuromass production and deposition via controlling proliferation patterns. And with this, I would like to end. And I would like to thank all the people involved in this study. So first, I would like to thank all these people who provided reagents. Then Andy is the graduate student who did all the migration and the deposition project. And Mark Lush, a postdoc, Rob Duncan, and Marina are working on the hair cell regeneration project. And I'd be happy to take any more questions.
Formation of the rosette structure of those cells, and you show it in your model as already a rosette, even in the anterior portion of the primordium. So is that organization already set up before it breaks off, or does it happen after it breaks off? And then is there a cell in the middle of the?
Yeah, so you mean here, this particular rosette?
Yeah, but then you show rosettes in the other more anterior portions.
Here?
So it's already set up there.
Yeah, so they do start to form here. And if you look at hair cell markers, they start like atonal comes on already at this stage. We also know that there is a central cell, and it's probably an FGF secreting cell, which is important for making these rosettes. And that's why you lose rosette formation if you get rid of FGF expression by these cells. And of course, as you can imagine, notch signaling is involved here in making these rosettes, hair cell versus support cells as well. And other groups have now shown that there's probably feedback between notch and FGF signaling here in the back. So the Wnt FGF interactions seem to change as you move the rosettes out of the tip region, and the control starts to change. Yeah.
Do you think there's a cross gradient as well?
From dorsal to ventral? We have not detected one so far, no. What do you have in mind? Do you have?
Well, I'm completely affected by listening to Eddie.
Well, that's where you made your first mistake.
Yeah, so we don't think so.
Can you explain it simply by the gradient of Wnt?
Yeah, so if we look at the morphology, we only do have an AP axis. We don't really have a DV axis to define here. And the primordium like in the movie where I showed that the primordium can turn around, it does change DV, and it apparently doesn't mind, right? It actually starts depositing sensory organs normally, irrespective of which direction and whether it flipped its DV axis. Yeah.
In one of the early slides, you had a black line indicated as periderm. Is this a layer? What holds all this group of cells together? Is it that they all sense the same things and they walk along? No. Or is there an enclosure, a sausage casing somewhere?
Yeah. So the primordium, I think itself the primordium holds together by cell-cell adhesion. So I think you could probably explant it, put it in a dish, and it would still migrate as a cohesive. And we have not determined yet what the signaling adhesion molecules are between the cells in the primordium. The periderm, the black lines that I indicated at the beginning, they're actually outside on the skin of the embryo. Yeah, they're on top. Yeah.
Is there a mutant scenario that we don't, I don't know if it's Piotrowski or whatever, that you don't actually form the rosettes and they keep proliferating, and what do they do? Do they deposit just non-differentiated cells? Because there's obviously proliferation and differentiation going on. Do you have other mutants that you can prevent the rosette formation yet still get it depositing cells?
Yeah, so we don't have a tool yet to dissociate the two. So if you inhibit rosette formation, the primordium doesn't migrate, because it's the FGF mutant that feeds back onto Wnt signaling, and then you mess up the chemokine signaling. We have mutants, so if you inhibit Wnt signaling, the primordium actually does migrate, but then it runs out of cells and it doesn't deposit anymore because it's not proliferating. So I think it's probably, I don't know. We can't really experimentally dissociate it, unfortunately.
You said the proliferation was really tied to this sort of directional movement as well. If they weren't proliferating, they started to tumble. You said tumble.
No. So this we don't know yet whether proliferation affects migration, but you need to migrate in order to get your sensory organs sort of normally spaced, and that's what I meant by coupling them, but. Not that one affects the other. Yeah.
How does this initial polarity get set up?
Good question. And we were hoping to see the same. Not hoping. We were looking whether we would see the same in the early primordium before it starts migrating, and it looks different. So left one is everywhere. FGF is actually the first rosette forms on the other side, on the posterior, and then the front grows, and then the whole thing takes off. So we don't know yet what polarizes the primordium before it starts to migrate. It could be signals from the hindbrain, or just body, some signals from the body axis, but we don't know yet.
And how many cells are in the initial grouping?
So with this transgenic line, you start maybe seeing 20 cells. I mean, then we start seeing them, right? You need a cluster of cells before you recognize them as a placode. And then they start to proliferate really rapidly. So first you see sort of a sensory organ, and then more proliferation, and then it takes off.
So this question is, I think, is pretty out there. But when you put a zebrafish embryo in colder water, I think development slows down, and it does so in pretty much a very coordinated way. And so, first part of the question is does the deposition of the sensory cells, does that get altered when you alter the temperature of the water?
So we have not looked yet, because we always assume that if you lower the temperature, you'll affect proliferation as well as migration speed. So you lower everything. So I think it would probably be normally spaced. So what we would like to do, I think what you're getting at, is try to affect migration speed and leave everything else the same, right?
Well, yeah, I assume that you can't do that by temperature. I mean, it would be interesting if you could, but yes, that's what I was getting at.
Yeah, so I think temperature will affect all these processes. It would be hard to dissect them.
How do you think that temperature can control every cell in development at the same time? Like, do you have any idea what the mechanism of that is? Is there anything known about that?
No, I'm not a biochemist, and I always just assumed that all biochemical metabolic processes would be lowered or raised at the same rate depending on the temperature, but I might be wrong. Yeah.
In your initial diagram, you showed the afferent nerve, right, that innervates the hair cells. So the hair cells are not really neurons, right? They are secondary sensory neurons, which are sitting, well, that's part of the question. They're sitting near the hindbrain, and then they send out long fibers. The question is, do they follow simply this trail of the primordium, or do they grow at the same time?
Yeah, so this has been described actually by Darren Gilmore at the EMBL. So what was known before is that the placode, before it starts to migrate, it actually leaves behind some cells posteriorly, which will form the ganglion. So they stay behind. The anterior part of the primordium starts to migrate, and then these ganglion cells will grow out axons, which will interdigitate. So the growth cones are in the primordium as it migrates, and then they start to innervate these sensory hair cells. In addition, new axons are also growing after the primordium has migrated, and will innervate more hair cells as the sensory organs grow. But so in contrast to the situation in the brain, here the axons follow these lateral line cells, and then there are also glial cells which then follow the axons.
And can one delete, say, the neurons, and the migration of the primordium is still okay? So
they migrate normally. Yeah, first in the back.
I guess there's a lateral line on each side, and so there's two primordia on each side, right? Do they deposit at exactly the same spot on both? Because I thought I saw one.
Yeah, no, they don't. I mean, if you look from a dorsal view, you can see that there is noise in the system, in the sense that they can be a few somites apart, and the sensory organs are never in parallel.
And that's because would you argue that that's because the proliferation rate on each is a little different? Or it starts at a slightly different time?
Yeah, I don't know whether. So I think proliferation usually introduces noise, at least in somatogenesis, they say that. I think they do start to migrate simultaneously, but then it becomes noisier and noisier.
But that doesn't mean anything for the fish?
No.
In terms of the staggered?
No. And 2 weeks later, they will have sensory organs on every somite. So then at that point, they're symmetric again. Yeah.
Okay.
Yeah.
Yeah, I had a question sort of related to Michael's about DV. So there is DV in the patterning, right, in terms of the orientation of the hair cells?
So what I didn't tell you is that there are actually two primordia migrating. So there's the first one, which I've shown you, and then a day later there's a second one coming. The first one makes sensory organs where the hair cells are all oriented along the axis of the embryo. So they will either respond to water flow coming this way or this way. Now the second primordium makes sensory organs where all the hair cells are oriented along this axis. And so, but with
Different primordium.
different primordium, yeah. So essentially, the organs coming from this particular primordium will all have the same axis.
So they'll all be along AP, and the second primordium will all be DB.
Yeah, and I think it's not quite understood yet why the second one, how the second one makes them orthogonal oriented. Yeah. Somebody in the back too? No? Okay, yeah.
Yeah, I'm really curious about how that organ is kind of put together. So it seems like you have a primordial stem cell and then a later tail that differentiates, and that FGF is really critical for the rosette formation. So does that mean that the primordia doesn't really have any FGF producing cells, and if you were to make that happen, you would induce that differentiation in the primordia? So
you mean in the leading region?
I guess,
or?
yeah, what I'm trying to get at is what is so critical about the distinction between these two zones, the differentiation zone and the primordial zone that is migrating.
So I guess it's you want FGF signaling in the back to make the rosettes there. So the question is, why do you not want it in the tip? And what we find experimentally is that as soon as FGF invades or becomes activated at high levels in the leading zone, you make a rosette there as well. And so these primordia do not migrate anymore. And I assume they would also, we don't know, we have looked at proliferation rates, but they do not migrate anymore. So they, at this point, we cannot study then deposition anymore because the primordium stops migrating. But I mean, this is now hypothetical. I assume that if you differentiate your cells in the leading region and you lose also your hypothetical stem cell compartment, the primordium would not make enough cells to really make it towards the tail.
Do you have a particular proliferating zone that is leaving the primordia out of? I mean, so where is that proliferation taking place?
So in the anterior 2/3 of the primordium, even in the tip as well as posteriorly. So based on proliferation patterns, we were not able to identify stem cells just by looking at proliferation. And I mean, we haven't looked yet at cell cycle lengths that might maybe give us an indication. But based on the mitotic index, we don't see a compartment that behaves differently than any other compartment.
Can you take this and cut it in half, or say, suck out the posterior part? Will it regenerate the whole again? Will it self-regulate?
Yeah, so people anecdotally have told me that they've done these experiments, but it has not been published yet. So I don't know whether they completely regenerate or not. I think they do not. So they don't upregulate proliferation. They keep on proliferating at the same rate. And I think at some point they become too small and cannot maintain this polarity, and then they stop migrating. Yeah.
You may have shown this before, but can you measure, just use indirect immunofluorescence to see whether there is a Wnt gradient?
We've tried that, yeah, and we haven't tried it exhaustively, but we do see it. And actually what we found recently is that even beta-catenin transcription makes a gradient. So, and this is interesting, yeah, that it's actually regulated by Wnt signaling probably.
And could that gradient be vertical rather than horizontal?
We haven't, so far we haven't seen any genes making a gradient dorsoventrally, no.
And what would set up a Wnt gradient? And I know you don't know, but I'm just curious.
So I mean, we know based on this Wnt10A expression that the Wnt expressing cells are in the tip, and then we think that it's then diffusing posteriorly, and we have evidence now that heparan sulfates, so proteoglycans, extracellular matrix proteins are also involved in the diffusion of that Wnt signal. So if we manipulate these proteoglycans, we now can expand Wnt expression or Wnt target gene expression throughout the entire primordium. And the other reason why we think it's a gradient, if you look at different Wnt target genes, so for example, if you look at NODEM, which has been described in fly imaginal discs to be only activated by really high Wnt signaling, we see it only in a few cells in the tip. Then you look at LEF1, which is in a few more cells, and you look at FGF10, which is another Wnt target, which now is activated in even further, in a wider domain. I mean, you could argue that this is different affinities for, must be, right, for the Wnt ligand. But I mean, just based on the fact that nodum, which has been described to be only responding to high levels of Wnt signaling, that is really restricted to the tip, makes me think that this is where you see the highest Wnt signal activation. You don't seem convinced.
Dickoff must contribute to the gradient too. You have Wnt and immediately behind it Dickoff. So. Yeah.
And we think that Dickoff might actually, in addition to only inhibiting Wnt signaling, the canonical Wnt signaling pathway by binding to LRPs, it might actually also bind to these heparan sulfates, which has been shown in fish before, and the signal via non-canonical Wnt signaling, which then inhibits the canonical. So I think it's more complicated than we think so far.
If there are no other questions, thank you very much.
Thank you.